Perfect. So now we're recording. We were discussing before we started recording, that's why we're a little late, the lab practical tree. There seems to be some issue with it, and I'll probably just have to rebuild it completely. So you'll see that coming up later tonight, and then I'll put 2 weeks from today for getting that done. I'm sorry about that. I thought I already once fixed it, but it seems to be stuck on something that's happened while it's being built, and I shamelessly just stole that practical from someone else. So I can't figure out what's going on with that building on that. But this week, we are working on the digestive system. It's week 13. And there's a bunch of things that I wanted to cover today, in addition to the actual chapter review. And of course, this takes us to the first one, where I'm going to have to make a change because of everything that's going on. I'm going to remove that practical tree. Practical tree will have 2 weeks from now to finish. So I'll send a notice of that to the entire class. But I want to talk a little bit about the exam tree. Exam 2 and practical 2, most students should have all of those back. We'll look at this week's module. There's going to be a lot of anatomy. It's pretty anatomy heavy, which I think actually will make it easier. We'll look at what we're supposed to do this week, and then discuss a little bit of the kind of general stuff that we've been having a look most of the sessions as well. And there's a little bit that I want to mention about the finals project, but we'll fully go through the briefing, and the grading matrix for that will be available for you next week. So, exam 3, this week is the last week to finish that. There's nothing new, nothing has changed from how you do those before, have done those before. 3 chapters, main emphasis on the respiratory system. In terms of the urinary system, the emphasis is then on the structure called nephron. There's a little bit about the water electrolyte and pH balance, but not huge amount. And this was fully the lecture content and fully from the study guide. So that should be easy. In terms of the practical, I'm going to have to confirm what's happening with that, but we're going to do it one way or another. It's just a matter of maybe it's under a different name than what you're expecting, how you're going to be completing that. So we'll get that sorted. Let me just write. So exam 3, try to get it done if you haven't had a chance to do it yet. I think you had until end of the day to complete the week 10 and 11. If you need extension for that, you let me know. If you need extension for exam, or if you need extension for anything else, you just let me know and we'll work from there. In terms of the exam 2 and practical 2, exam 2, everyone should have their exams graded. Practical 2, almost everyone should have their grades released. I think I have 6 more students that I want to review for the 4th time, the practical 2. So most of the class has that also available. If you're one of those who doesn't have any comments there, then I'm going to release them tonight. So both of those scores should be available for you. In terms of a couple of general points just to make, always try to answer something. There were a few students, or quite a few students, who didn't answer anything when they were like totally at lost. It's better to just guess if it's an exam and it's multiple choice. Most of the time, multiple choices on my exams have 4 answers, so you're going to get on 25% likelihood that right. If it's a true-false statement, you have 50-50 chance to get it right. So at least guess something, especially if it's something that doesn't require you to type. If you have time, it really just takes a click. If you have no idea, at least try it. If it's question On a practical where you have to type in the answers and you have no idea, tell me at what location of the body we are. Tell me the cavity, tell me structure close to it, tell me something so I can give you partial points for it. I give points if I know that student has no clue, but they've given me something. You might not get full points, but you'll get some points. So I do encourage you to try to answer something at all. Something at least, so you'll get points for that. We also all have noticed that how the system automatically grades you is really, really harsh. So don't let that throw you off initially when you get your score. I'm going to have to go through them manually, and I think that I go through every single answer that you've given to every single question at least 4 times. So I'm pending for the 4th time for those 6 students on the practical too. But you'll notice that your grade changes significantly. I would encourage you to read the feedback. There's a general feedback, and then there's feedback on most questions. Usually if you've gotten something wrong, there's the feedback, and I discuss why you got it wrong or what you might use as a mnemonic or so on. So I encourage you to review those. If you don't have any comments in your exam or in your practical, then you haven't gotten that feedback yet. So just wait until end of today. You will have your feedback there and the score is updated. If you ran out of time, that happened for few students. That's not unusual. These tests, I think, are much more about time management than it is really about anything else. And I don't know if I fully agree with that. So for the next semester, we might change some things around. But if you run out of time and you're really feeling that, or the answers just didn't work out, something went totally not the way you wanted it to go, let me know. We can always reset the test. We can always figure out how are we going to help you to succeed. And the big thing that I do want to say, it's on a different font because I forgot it in this morning's session that what I wanted to say. I just had a bullet point. But always attempt the extra credit question if you have time in the exams. On exam 3, I don't think we had an extra credit question because I simply didn't have time to think of a good one. So we'll have a look at the end of the semester where you're standing, and maybe I'll put an extra credit question at that time. Well, we'll see where you're at, and I'll talk about that in a second. But what I did add for this evening's session, what I do want you to also pay attention, that check your grades, check the feedback, the comments that I put there. If I say that this question you had a right answer and then you still haven't gotten points for it, let me know. I do sometimes mistakes. It's especially since I'm looking 50 questions times 30 students times 4 times. That's reading it through so many times that sometimes eyes might jump over a line. So do let me know if you don't agree with the score that you've gotten. It doesn't align with the written feedback that you've gotten. And this is true especially for the practical exams. But most of the time I'm correct, but it is possible that I'm wrong. So if that's the case, don't hesitate to let me know. This week, I'm going to talk about this week's assignments in a second, but from last week, the critical thinking, some students were having issues with that, and not because of the students, because of the way it was set up. So I'll have to look into that a little bit more, and then you'll get your grades back. There's something that I need to make sure that everyone gets treated the same. So bear with the critical thinking question feedback. This week, what we're doing is very similar to other weeks. We are doing the assignments. Only difference in those is that you have 2 labs. So make sure to do both of those labs. Nothing else is really unusual from what you've done before. And I think that all of these are through the McGraw-Hill's Connect. So it should be fairly easy and fairly logical to work through. We're going to go through the key points of the digestive system, but just in case if someone needs to head off or there's other things that pop up, I do want to show you a couple of points. This one we've already seen before, that if your Connect access code has expired, college says that you need to purchase a new one, but I realized that at this point of the semester, no one wants to buy a new access code because we're almost done. So I am working to get the alternative assignments for those. But in all honesty, I have most of them ready, but I need to still work on some. So if you are needing alternative assignments, message me through Connect, so I know also not to work on an assignment that no one's going to do. So if your Connect code no longer works, there is an alternative way of doing this. Of course, you can purchase the new access code, but the alternative assignments are something that's more of a Word-based document you complete for me. Also, in terms of that, I have opened rest of the semester's assignments, rest of the semester's modules for you. So if you are noticing that your Connect access code is going to expire in the next few days or next week or so, you can actually work ahead and get all of those Connect components completed. So we won't be using Connect on your finals week. So if you feel that you really want to get those out of the ways and you're worried that your Connect access code expires, they are available there for you to do. They're all open. And then I wanted to say a couple of words about the missing assignments, but also that this applies to the exams and practicals. If you have missed one, or if you did totally bomb in one, you just have to let me know, and we can figure out if I reopen that for you, if I reset it for you. So whether it's an assignment, whether it's an exam, or whether it's a practical, I don't want you to feel that you really didn't do well on one and you would really want to attempt it again. If that's the case, I'll work with you and we'll have a look if that makes sense that you reattempted. If it's very close to the end of the semester when you end up noticing this, then it might be that it's not a good idea to reattempt it. It might be a better idea to do extra credit activity. In terms of the finals project, this is again just to put the bug in your ear next week. I think I have the grading matrix available for you. So I want you to see how I'm grading you before you do your finals activity. And this is something that you'll be completing during the last week. It's going to be very small in workload. I don't want you to work like crazy. I know you have other courses, other assignments to do during the finals week. But it's a project, and it's going to be an independent project that you complete. It can be, for example, about a clinical condition that relates to the things we've discussed. It can be a topic that we've discussed. It can be how different systems work together, or something that relates to what we've talked and you're interested about. Some students have preferred to do posters on a PowerPoint slide. That can be kind of a nice one. You just collect the information, make it nice. Some students have preferred to write essays. Some have preferred to give a presentation. You can do many different things. But the point is that I'll give you the details, how I'm grading it, and then you can figure out how you get those points. And there's going to be also extra credits available during the finals project. So you can do what's needed and get the full points, or you can do what's needed and a little extra and you'll get extra points. So that's the chance where you can look where you're standing with your grade and how much more do you need that you get the grade that you're after. So really the big thing during the finals week is that I'm trying to make sure that I have time with everyone and that you have time, everyone has time, either to interact with me through a message or preferably doing one of these web meetings so we can look at exactly all the points that you've gotten in your assignments, that you are happy, you agree with what's recorded, but also where you are standing with your total grade, and is it what you need, for example, to a program where you want to get. If it's not what you need, for example, for a program that you want to get, then you let me know and we'll see what else can you do to get there. Of course, if you're like standing at the D and you need an A and it's the last week of the semester, that might be challenging to make that move. That might be a little bit too much. But if you're standing, for example, at the C, you want to get a B, or you're standing at B, you want to get an A, it's probably likely that you will be able to do that as long as you put in the effort to do the extra assignment, whatever it will be. Also, do remember the tutoring your college offers that. I also am happy to do tutoring. But that's all the stuff regarding that's kind of a new material. Do you have any questions about those? If you do, I'm happy to address them now. I'm happy to also address them later on at the end of this session. Otherwise, if there's no questions, I'm just going to jump in to talk about the digestive system, what we need to know about this week's material. But like we've discussed already, the big thing is that the practical tree, there's something wrong with it, and I'll need to probably just rebuild it altogether. So don't feel that it's your fault or there's an issue with it. I'm also toying with an idea. If I can't get it rebuilt, it might be that all students just get the full points automatically. But I'm not making a promise of that. I'll need to think, what's the issue? Why isn't it working? So you'll hear from me by the end of today, by tomorrow morning, that what we're doing with the practical tree. Any questions, or are we good to jump into the digestive system and what's the key points of knowing about that? I know that it's kind of weird having all of those modules open now all at once, but it doesn't mean that you would have to rush. We're still going to keep with the same pattern of the material that's marked week by week. So I'm not expecting you to work ahead unless you want to. Well, I'm not hearing or seeing anything showing up on the chat that you would want to ask questions. But if you have, you can ask now, you can ask later on. I can also pause the recording, whatever feels kind of most useful for you. But I'm gonna now open up the PowerPoint for this week's material. And it's not done on the layout that I normally use for your slides. So look past that. Your recorded lectures, the lecture videos that cover the full of the material, not just the review, they will have the pretty format that your college uses. These are on another format or from another college where I work. You can work it out based on the colors, but it doesn't change the content. We're still covering the same content. The chapter numbers might be off as well. So this week we move on to the digestive system. And on this first part, we're just going to talk a little bit about the general concepts. And then we're going to focus on the oral anatomy. We have quite a few students who were wanting to go and pursue a career in that field, whether it's dentistry or for some other reason, looking at the digestive system, especially the oral system. So we're spending a little bit of more time than on other parts on the oral anatomy. Like I said, this chapter is heavily anatomy focused. That actually might make it easier for many of you. I find it much easier because things that we're looking on this chapter are, for the most part, concrete things that you can touch, you can feel, you can see with naked eye. There's a little bit of physiology, but really it's much lesser than on other chapters. So many students find this chapter much easier to cover. If we look at the functions of the digestive system, there's really four main things that we need to see. We're taking in food to the body, and then we're breaking that food into smaller components. And once they're small enough, we're able to absorb them. So we're able to let those cross the membrane between, for example, intestines and inside of your body. So that's when the material actually gets into the body and gets used there. Usually it enters the bloodstream. And then whatever is left that we cannot use in your body, we have to get rid of it as well. So those are the four functions, and we'll come back to them a little bit more, but just kind of to get you on a map of why do we have digestive system. And this diagram you've seen before, so there's nothing new, nothing surprising to you. I'm really not expecting you to memorize it, but what you'll notice is that this week we are focusing on this part of the diagram. So what we're doing, we're looking what your digestive system does. And if you really simplify things, your digestive system breaks down sugars and uses oxygen to that process. And as a result of that, we get energy. We get ATP. And then we have two waste products. We have carbon dioxide and water. And those are the ones that we got rid of, both with respiration and with the urinary system. So those are the two systems that we've already had a look on. So if you're a person who likes to go in an organized manner through substance or through chapters, it seems like we're jumping a little bit backwards in the material that we've covered. But it was really important to cover the respiratory system and urinary system together as a bundle because there's so much link between the two. So hopefully it doesn't confuse you too much. You'll see just that digestive system is an independent system on its own, but it has these links with the other material that you have seen already on this course. That's really what the digestive system does, as we mentioned. Breaks down sugars with oxygen to do ATP, which is our energy. And then we are left with two waste products. But like we already discussed, the functions of the digestive system is really to take in the food, break it into smaller, and then absorb it. And whatever is left, we eliminate. And you remember about the breaking materials into smaller components. This is one of the concepts that I expect that you would have covered in one of your introductory bio classes. And what we learned there was that whenever you break down molecules into smaller components, as a result of that, you end up producing energy. And usually it was the ATP. So this process of breaking down complex molecules into simple molecules results in energy. And that's really the reason why we are doing this. And the ATP was the form of energy, the energy currency in the body that we're going to be using. So this is just kind of connecting to what you already know. And then we had to get rid of whatever we couldn't digest. So these are the two important terms that are going to be introduced to you. What is the term that we use for breaking down molecules? And what is the term that we use when we actually take those smaller molecules, smaller components into them? And these terms are digestion and absorption. And I really would like to make sure that you're very, very clear and very comfortable that what's the difference between the two. That would make a fantastic exam question, I think. And this is really fundamental in terms of this chapter to be clear about these two. So digestion is when we take the food that we take into the body, and we take the molecules that are in that food that we take into the body, and we break them into smaller. So that's what digestion does. It breaks the molecules into smaller components. And the point of doing that is that they need to be small enough so we can absorb them. We cannot absorb big molecules. They need to be broken down to be very, very small before we can absorb them. So digestion was this process of breaking down whatever we've eaten and breaking it so small that we're able to absorb it. And absorption refers to the fact when these small molecules cross the membrane and end up into your body. And what do I mean by the fact that they end up into your body? When you eat something, isn't it inside you? Yeah, technically, but technically not. Because what digestive tract is, it's just a long tube starting from the mouth and ending up to the other end. And it's a continuous tube that expands and shrinks at different parts. But really, if you consider that what's outside us is outside our body, well, inside the digestive tract is also continuous with this space outside our body. So really, the only time when it really per se enters to the body is when it crosses that membrane of the wall of the digestive tract. So that event of crossing that membrane is the absorption part. So be very clear with what's the difference between digestion and absorption. So I just want to remind you of that, and that would be an important concept. We do have one more exam, which is going to include the digestive system. So I'm already highlighting that this would make a fantastic exam question. There's two types of movements that we end up finding happening in digestive system. And you've already heard me using the term peristalsis quite a few times. But now it's the time when we're really going to sit down and study what is that movement. And that's important to know because we use that term so much in A and B. You'll keep seeing it being used later also if you continue your studies. So the way I often explain this to my students is to think of this. There's two types of people in the world. And these two types of people can be classified based on how they squeeze their toothpaste tube. Especially if you moved in with someone, you live with someone else, you'll notice that if they're not your type of person, you'll be able to tell. Well, some people prefer to take their toothpaste tube and squeeze it from the very end and slowly move towards the tip where stuff comes out so that you get all the material out from there. They don't just go and grab it from the middle. They work from the end towards the tip of the toothpaste tube. And these people, what they're doing, they show the action of peristalsis. In peristalsis, we have this tube that's a muscular tube, and the tube contracts starting from one end, and then it contracts right next to where it contracted and continues in this wave-like movement that helps us to move that material within this tube ahead. It helps us to make sure that the material moves, and it moves only to one direction, thanks to the manner on which this muscular tube contracts and generates almost like this wave that helps the material to move down the tube to whatever direction that tube is going. Well, that's one way, but then there's other people who go and grab the toothpaste from the middle and squeeze it from there. Yes, some of it will come out of there, but over time, if you always grab it from middle, eventually nothing will come out and there's toothpaste on both ends. They've just been squeezing from the middle. Well, these people would be doing what's known as segmentation. In segmentation, when we have this tube of muscle in a digestive system, it's not really moving the food inside that tube to any direction. All it does is that it contracts in some random part of the tube, and then it stops contracting. That doesn't generate this wave-like movement that would move materials along this tube. All it does is that it can break down food that's right at that spot that you contract, and it also helps to mix food that's right at that area where it contracts. It doesn't help to move the food along, it simply helps to break and mix the food within that tube. So I hope that those two make sense. If not, give me a shout out because this is an important concept to know. But peristalsis really serves the purpose of moving materials along. Segmentation, simply mixing and breaking food or breaking materials inside that tube. So that's kind of the general stuff. And now we're ready to have a look at the digestive tract in more detail. And really, when I think of the digestive tract, I think that it's about 8 meters long. That's a long distance. And most of it is packed into your abdominal region and kind of goes wiggling, squeezed in there. But all it is, it's a tube that starts at the mouth, ends to the other end. And most of the tube is very, very similar in the structure of its wall. Yes, at certain parts, that tube grows bigger. At certain parts, that tube goes smaller. In certain parts, the tube is just straight line. In other parts, it gets all wiggly and packed into the small space. But by large, the walls of that tube are similar. We find 4 similar structures in all parts of the digestive tract for that wall. Of course, then we have a bunch of accessory organs like our salivary glands, our liver, our gallbladder, and our pancreas. We're not going to spend huge amount of time talking about those accessory organs, but they, of course, secrete materials into this tube. Our focus on this chapter will be this tube. And then whatever is secreted to there it's important to know, but it's not as important to know as what this tube is. So we're going to have a look of this tube. And on this tube, like I said, we end up finding 4 layers in all parts of this tube. Most of the time, we find 4. There's one exception where we actually find 5th layer, but we'll talk about that, where we find within the 3rd layer 3 different sublayers. But when we look at the wall of the digestive tract, and we start from the center of the tube, so that's called lumen. And I think that I've discussed what lumen is. It's the center of any tube. With you guys already earlier on, I had one class where I forgot to do that, and students had no idea what I was referring to. So we're moving through these structures of digestive tract wall, starting from the inside the tube, and then we move slowly further and further and further out. So let's have a look of these 4 layers that we have. These are also sometimes referred as tunicas, and we've already heard the word tunica. That meant a layer, if you want to simplify and translate it in a very, very simple manner. So let's start with the innermost layer, the layer 1 based on this diagram. That is known as mucosa. Mucosa, as you notice, has a lot of these bumps and a little bit of structures that come out of the smooth surface. And that tells us a little bit about the function of this part of the digestive tract as well. And of course, function explains the structure. So the function of the mucosal layer is really to protect the rest of the layers. So it serves a kind of a buffer. So for example, if you have food that's very dry, let's say that you're eating cereal, and that's dry cereal that you would eat as a breakfast normally with milk. If you don't take any milk and you swallow it, it's rough on this digestive tube. So what the mucosal layer does is protects other layers underneath it. It's the one that guards that nothing else gets damaged. In addition, also, if you would, for example, have that dry food, we want to secrete stuff into that. So we have various different kinds of glands. These glands are not located within mucosal layer. These glands are located further within the wall, but the glands secrete their secretions to the mucosal layer from the top of the, or from the outer boundary of the mucosal layer. Also, mucosal layer allows materials to go inside the body, so it absorbs materials. So it's very good at doing 3 of these functions. Let's have a look at the next layer, and the name of it tells where it's located. It's submucosa, and if it's sub, it means underneath or below the mucosal layer. This layer contains a lot of glands, a lot of blood vessels, a lot of nerves. So really the function of having all these blood vessels, nerves, and glands is that this layer helps to regulate what happens with the digestion. So the glands are located within the submucosa. They open up their ducts at the mucosal layer. The nerves and the blood vessels are able to tell us that if we need to do something more or if we need to do something less. So this submucosal layer is really the regulatory layer. It observes that is everything going the way we want, and if it's not going, what do we change? So that's the function of the submucosa. The third layer, which is pink in this diagram, and you'll notice it's quite a thick layer, is the muscular layer. And this muscle is smooth muscle. If you remember our review of different muscle types from the very beginning of BIO201, we have different kinds of muscles, and smooth muscle was found in the digestive tract, but also around blood vessels. We've talked about that a little bit. And this muscle is not all the same. We've got 2 different layers there, 2 to be precise. We've got an inner layer, which is circular. So it runs, if this is the tube, it runs around that tube as a circle. That's the innermost layer that we see here. The outer layer of the muscular layer of the digestive tract wall is actually longitudinal, so it runs the fibers along that tube that we saw. And it cannot really squeeze that tube, but it can kind of bring the length of the tube back and forth. It can make the tube longer or shorter. There is one exception to this. In stomach, we find the third kind of a muscle layer. And this is oblique fibers. So they don't run into any direction that we know in terms of X and Y axis. They run in something in between those. And the function of having a third layer within the stomach, and we'll talk about that a little bit, is that stomach really needs to mix up stuff. And stomach needs to be able to stretch and shrink. So that's why we have a third layer there. And then the very last layer that we find in the general model of the digestive tract is shown here, and it's quite thin, the number 4. That's just connective tissue, and its function is to anchor the digestive tract to its location. So that's all it does in this simplified revision that we're going to do. It just keeps the stuff on its place where it needs to be. So those are the general layers of the digestive tract. And we'll come back to these. I'll show an example of these a little bit later. But at this point, I want to discuss about one structure. And this structure is a structure that we've heard a lot already in the past, but we have never really looked into great amounts of that. You should have seen it when you covered on BIO201, the cavities of the body. If you didn't, you'll see it now. This structure is known as peritoneum. So peritoneum is simply serous membrane that lines all of the abdominal cavity. And I have here a very nice diagram that I'm going to use to highlight. So we'll see a human body cut in half, right and left. We'll see this is the posterior part, this is the anterior part. You'll see we've got our vertebral column running here, and our belly would be here, a belly button here. This is a female body, so you'll see female reproductive tract there as well. But all of this abdominopelvic cavity is lined by this red line that turns blue in certain parts, but it's still the same part of the same structure. And this line is known as peritoneum. So inside this line, we find the peritoneal cavity. Everything that's yellow is part of this peritoneal cavity. You'll also notice that at certain parts, this peritoneum goes around structures that we find within the abdominopelvic cavity. We go around the liver, we go around stomach, we go around small and large intestines. So the function of it is, there's a few different functions. First of all, we want to form this kind of sac. So infection, for example, cannot spread from outside the peritoneal cavity inside the peritoneum. Also, an infection within the peritoneum cannot spread to other parts of the body. In addition, this, like we discussed, anchored structures on its place. And we'll look at one of these loops that we see here. And the loop that we're going to look at here is this loop here. This loop gives us a structure known as greater omentum. So when you dissect, and I really was hoping to take this class to actually see a dissection lab, but because of the COVID, we're not going to do that. But when you dissect a person and you open the stomach, you open the abdominal cavity, you remove the skin, you remove the fat tissue, you remove the muscles that are there. This is the next structure that you will see, this kind of a flap-like loop of serous membrane. You'll find a little bit of fat, adipose tissue on it. You'll find blood vessels running. That's the greater omentum. So that's the reason why you need to know the structure, that this is the first one that you'll end up seeing if you actually cut into the human body. So that's probably all that I wanted to say about the peritoneum. I do want to remind you, we've discussed that certain structures, how they are located in relation to the peritoneum. For example, kidneys, how they were retroperitoneal, so they're behind the peritoneum. You can have structures that are intraperitoneal, so they're inside the peritoneal cavity. And then you can have, for example, subperitoneal organs such as your bladder and so on. So below the peritoneum. That's probably all I want to say on this review about peritoneum. We'll go into greater detail on the actual lecture videos, but for this review, I think that covers it. In terms of neural supply, and this is just kind of a reference to what you have discussed at the end of the BIO201, just to remind you, our autonomic nervous system had two branches. We had the parasympathetic and sympathetic branch, and our digestive system is acting within the parasympathetic branch being active. So when you are doing what was known as the rest and digest, so your parasympathetic branch is active, that's when your digestive system is working. This is the time when you are, let's say that you've had your Thanksgiving meal, since Thanksgiving is coming, and after that you sit down. You're not moving about, you're not worried that someone's gonna attack you or kill you, you're not working hard, you're just chilling on that sofa or chair or whatever you're sitting at the end of the Thanksgiving meal. That's when your parasympathetic system is active. You'll notice that your heart rate goes down, you're breathing slower, but deeper as well. Your pupils are small, you're relaxed. That's when your digestive system is active. When you are in a situation where you're, for example, worried about being attacked by a lion, that's when your sympathetic nervous system would be active. You're only worried about staying alive then, and in that immediate time. Your heart starts to pump faster, you breathe shallow, fast, your pupils are very expanded. Your body is ready to either fight against that threat or run. So flight or fight responses. That's when your digestive system goes completely down. We don't spend time, we don't spend resources on that. Because if you're going to die, it doesn't matter whether you digested that food or not. So remember that it's the parasympathetic branch of the autonomic nervous system that's active when we're doing digest. So just kind of a quick reminder. Other systems that would be very active when the parasympathetic nervous system is activated, your urinary system as well. The other thing that I want to say, and this is not the most beautiful drawing, but it's the best I could find, this is looking at the abdominal cavity and showing our large intestines, our small intestines. And what I want to show with this is that there's a lot of blood supply to our abdominal cavity and to supplying the digestive system. So there's a rich blood supply to the intestines, and there is a reason for this. Because this blood that comes to the digestive system, comes to the intestines, picks up all those nutrients, all that energy that manages to be absorbed, so taken across that membrane. So all that is picked up by the blood in that area. And this blood, for the most part, gets directed to liver. And why it goes to liver is actually quite a smart mechanism in the human body, because in general your tissues, your cells in the body are greedy and they're totally irresponsible. If you give various tissues in the body a lot of nutrients and energy, they just go crazy and burn that off, and then that energy is gone. Liver is one of the few organs that approaches extra energy in a responsible manner. What liver does, when all of that blood from the intestines comes to the liver, it collects the energy from that blood and stores it for later use when you might need it. So the blood that has gone through the liver and then goes to the rest of the body doesn't have that much energy anymore. We store most of the energy that we are able to pick up from the intestines with the blood. We store it to the liver. So that's the blood flow that you need to know about the intestines and digestive tract in general. Well, the next thing that we need to look is, this is kind of the introduction. The next thing that we need to look is, where does the digestion start? Many students answer to me that, oh, at mouth. And once you put something to the mouth, you start digesting. And I think they're close to the truth. So when you put food to your mouth, you start tasting it, you start breaking down, for example, starches with the salivary amylase in your saliva. But I don't know about you, but when I see food that I like, just visually, or when I smell food that I like, and especially when I taste food that I like, I already start salivating. The food doesn't have to be in my mouth for me to start salivating. So really, these digestive processes start even before the food enters your body. Just the mere sight or smell of the food can trigger your digestive processes. And saliva was probably the best example of this. Pavlov's dogs, if you have studied psychology, they started salivating just when they heard the sound of the bell, because they had been conditioned to connect the sound of the bell to the fact that they were going to get food. So digestion starts even before food enters your mouth. Just this mere sight or smell starts us to produce saliva. And saliva contained one of the digestive enzymes that we'll be looking on this chapter, and that was the salivary amylase, which broke down starches. So there's a nice picture of saliva. I don't know where I put it, but I did. So I spent a lot of my time in the teaching in one of the dental schools. So we will spend quite a bit of time on mouth, and then we'll cover the rest of the digestive tract. But especially knowing that in this class we have students who are heading to a dentistry field or fields like that, we are going to focus on the mouth a little bit more than the other parts. So mouth is important structure for many reasons. Well, first of all, that we've already seen, taking in food. We actually screen the food that we take in as well, if you think. We taste it. Does it taste okay? We might physically feel it with our tongue. Does the texture seem okay? Does it smell funky? Does it look weird? All of those things happen even before the food enters to our mouth. So we screen that, is this something that we should be eating? And then if we agree with that, that's where the food gets into our body. So two important functions of the mouth. The third one, which we have already seen in our previous chapter of the respiratory system, of course, at the level of the mouth, we manipulated that airstream that was vibrating thanks to going through our vocal cords. So speech is formed largely by manipulating that airstream at the level of the mouth. So communication is another important aspect of the mouth cavity. And you can see two terms being used other than mouth cavity quite a lot. You can see a term oral cavity, and I think that we've already used that in the past. But another term that you can see sometimes being used is our buccal cavity. So that's another term that refers to the same thing, mouth cavity, oral cavity. So don't get thrown off if you see either one of those being used instead of mouth cavity. And I think that always when we start studying a structure, it's a good idea to define the borders. What are the boundaries of that area? And let's look at the area of the mouth. What sort of boundaries we have? And if this was more light class, I probably would be trying to quiz you. But now, for the interest of time and interest of the lack of interaction, we're just going to go through these together. So most anteriorly at the front of the mouth, we notice we have our lips. These prevent the food from falling out to the front of our mouth cavity. So anterior boundary of the mouth is formed by the lips. Laterally on the side, we have our cheeks that form the boundary of the mouth. Superiorly, so the top boundary of our mouth is formed by the soft and hard palate. And you notice that the hard palate was the more anterior part, and that's hard because there's bone actually inside this tissue. And the soft palate was the posterior part, and right at the end of the soft palate we had uvula hanging down. It just didn't fit into this diagram when I included it. So that's the superior, the top boundary of the mouth. Well, the bottom part of the mouth, the inferior boundary, is actually quite interestingly our tongue. And if you look at this diagram, this is not unreasonably drawn. This is pretty much to the scale. Your tongue is huge. It's not a single muscle. It's actually made of groups of muscles. And it's really large. And I realized that I was brought up and I've spent first years of my life in a very different society and a very different way of life than people here do. So eating tongue was quite a delicacy in that environment. Tongue is all muscle. There's very little anything else. So it's really fleshy and kind of enjoyable in many cultures, considered as good food. If you go to supermarket here, I think you can see cow tongues every now and then, at least if there's a butcher in that supermarket as well. So if you're interested, you can figure out yourself what you think about the tongue. Is it good food or not? What else? Posteriorly, at the back of the oral cavity, we have our oropharynx. That's something that we're already familiar. We've already talked about that in the past. So that should be a familiar term. There's one more term that I do want to introduce in terms of the boundaries or borders of the mouth. And this term is vestibule. Vestibule, if you translate it literally, it means chamber. And if you think about the term vestibule and to what area it's referring, the term chamber is actually a good description. So vestibule refers to this space that's between where your teeth and gums are and your lips. It forms like a little chamber between the teeth and gums and the lips and also to the cheek. So this area is known as vestibule, a chamber. So just a term for you to know as well. Let's have a look of tongue as the next thing. We saw that it's on the floor of the mouth. It can be divided in multiple muscle parts. It's not a single muscle. Really, the big thing that I want on this review session highlight for you, and rest of the detail you can find on the actual lecture videos, are these small rounded projections that are on the surface of the tongue. Some are smaller, some are larger. These are known as papillae. So those are important part of the tongue structure. There's a couple of more structures. I don't know if you've heard about these before, but if you haven't, these are definitely worth knowing. We have a structure known as frenulum. Frenulum is this connective tissue band or small fold or small ridge that you'll see underneath the tongue connecting to the gums inside the mouth. You'll see this little band here. So that's known as frenulum. And in fact, you've got various different frenulums in your mouth. You don't need to know more about them. You don't need to know the specific names. You just know that in general, they are known as frenulums. You also have another frenulum here at the front on the lower jaw, connecting from the gums at the very front to the lip. So there's another frenulum. There's actually a third frenulum also on the upper jaw, connecting the gums to your lips here. So if you have a good friend, you ask them to show those for you. If your friends aren't creepy like that, that they're willing to do that, you can go in front of the mirror and see those frenulums on yourself. The reason why I want to highlight those, not only that they're a good anatomical landmark, and we'll actually use this frenulum as a landmark to notice certain salivary ducts where they open, is that sometimes when babies have trouble with latching, we might end up having to cut a frenulum. So if that might be something that happens sometimes as well. So that's why I wanted to introduce those. Another one that we actually see at the back here, back of the soft palate, of course, are uvula, this hanging projection down from there. Its function is to regulate the sensory organs, so we notice if we swallow something or if we're about to swallow something and we're not ready for that. That kind of detects that. A lot of people, if you touch uvula, they get a gagging reflex if they're not prepared to swallow. Also, uvula raised up with the soft palate sealed off the nasal cavity, so that food doesn't go to the nasal cavity like it shouldn't. Here's just not something that you need to worry about. This is not in the exam, but I think it's a funny thing, or not a funny thing, but interesting thing. This is a different kind of uvula. We don't normally see these, but every now and then, if you look into the mouths of people enough, you'll end up seeing these. So this is a bifid uvula. It's a uvula that has been divided in two, and usually that's consequences of some issues having taken place during the embryonic and fetal development, where all parts of the face didn't fuse fully perfectly. It can be more serious, or it can be just a slight reminder that it didn't happen exactly like it was supposed to happen, but your body was able to catch up, but this part didn't get fully fused. So just to show you something, I really feel that when we're studying these general A&P courses, we always assume that everyone looks the same and everyone's structure is the same. Well, that's not the case. There's a lot of variation that we end up finding. So I try to show you some oddities every now and then as well. So this would be one of those. So now that we've talked about those, we kind of jump back to what we talked about. We talked about saliva earlier on, and saliva had many different functions. We started with noticing that that was one of the first digestive secretions that we made. Saliva contains salivary amylase, which was really important in breaking down the starches. In addition, saliva moistens the food. So think of if you eat a dry cereal, it would be hard to swallow it. But once you mix it with saliva, it actually becomes kind of nice. It lubricates that food and it's easy to swallow it down. It also can be used to clean mouth. This is what doing the chewing gum is all about. It makes you salivate a lot. So that cleans your mouth. If you don't have a chance to go and brush your teeth, you eat a chewing gum instead. One term that we end up noticing here, and I'll highlight this later on, so don't panic if it doesn't make sense, but I've talked so far that we eat food and then food travels inside the digestive system. That has been an oversimplification. We need to use correct terms. Once you mix food with saliva and you mechanically break it by chewing on that, we no longer talk in the field of anatomy and physiology or in the field of medicine, we no longer talk about food. Now that has turned into something that we call a bolus. Bolus is easy for me to remember. I kind of think, oh, it's like a ball-like structure, you swallow it. So that food, once it gets mixed with saliva, once it gets mechanically broken down, we start talking about bolus. So that was just kind of a side branch, but I do want you to be able to use the correct terms. So what do we need to know about saliva? That's a good start, but we need to know what produces the saliva. And we've got salivary glands that produce the saliva for us. And there's 3 ones that I want us to know. And we're going to go through them. Some of them you have on both sides, some of them you don't. We're going to go through them in order. And I want to start with this one here. And this one is known as parotid gland. So parotid salivary gland is located here next to the ear, slightly above the masseter, if you studied the muscles of mastication really well. And you'll notice that it has a duct, and this duct opens up next to the second molar on the upper jaw. Parotid salivary gland is probably the largest out of these 3. You've got one on each side. And Cranial nerve 5, the facial nerve, actually runs through it. So it's not unusual that we could have cancers of parotid salivary gland, and we would need to do surgery there. But doing the surgery is dangerous or difficult because that's where the facial nerve runs through. So that adds a level of challenge for us. How do I remember that this is parotid salivary gland? I think of pirates. Pirates tend to have a parrot sitting next to them. And where the parrot would be, that's where your parotid salivary gland is. So you might think of something smarter, but I'm all about thinking simple mnemonics. So that helps me to remember where the parotid salivary gland is. Let's look at next, submandibular salivary gland. And normally the name is a very good clue where they're located. I think this is a little bit misleading. It's not really underneath the mandible, it's more of medial to the mandible, so kind of inside it, in the middle of the lower jawbone. It's not significantly down. I guess that it could be slightly down. You can even feel your salivary glands if you're very imaginative. But really, they are below or within the mandible. You'll notice that they have a duct that opens at the base of lingual frenulum. And we just saw that here. These openings here at the base of lingual frenulum. These are where our submandibular salivary glands ducts open up. So this is why we learn some terms, because we use them as landmarks for then finding other structures. And the last one that we have to look at is our sublingual salivary gland. So that does what the name tells us. It's below the tongue. And it actually has many ducts or many opening points at the floor of the mouth. So I guess that one of the things that we should look at here is the secretions from these salivary glands. And not all salivary glands secrete the same stuff. There's a little bit of difference in what we're secreting. So let's have a look at those. And we're going to start with a secretion that's described as being serous secretion. So serous is watery, very liquidy secretion. Very fluid. When in comparison, we can have secretion that's more of a mucus secretion. And you remember from our previous chapter that mucus secretions are kind of thicker, like that stuff that you produce in nasal cavity that you might then sneeze out, that snort. So mucus secretion is much thicker than our serous secretions. So which gland secretes what kind of secretions? Well, parotid gland secretes serous secretions. So it's very watery secretions that we get from the parotid glands. And this mucus secretion is actually secreted from our sublingual salivary gland. So that secretes this very mucus secretion. Well, how about the submandibular gland? Well, secretion from the submandibular gland is kind of a mixture of serous and mucus secretions. So it would be worthwhile for the exam to know different salivary glands and what kind of secretions they produce. And often I ask from my students, how much do you salivate in a day? How much saliva do you produce? Well, it's actually more than most of us end up thinking. It's about 1.5 liters. So I think it's a little more than one of these cups. That would be about a liter here. So it's quite a bit of saliva that we produce. How come we don't become then dehydrated? Well, you just learned in the previous chapter about the fluid balance. You know that even though we salivate all this, it just doesn't go through us. We reabsorb a lot of this fluid back to the body. So that's what we need to know about the saliva and salivary glands. The next thing to talk about is going to be our teeth. And teeth are really some of the hardest structures in the body. It's simply because of what their function is. And we all know that between the 6 month and age of 2 years, you end up getting your primary teeth, your milk teeth, or some call them baby teeth. There's going to be 20 of those. And really, when the teeth emerge from the jawbone, where they develop first and then they come through, that can be quite painful. So babies end up crying. It ends up being a hard process that they go through. That's our primary teeth. Then at between ages of 6 to 12 years on average, you get your permanent teeth. So another set of teeth. And there's actually a little more of these because now our jaw is bigger, so we can fit in more there. There's 32 of those, and these are our permanent teeth. So they actually keep growing in the jawbone underneath the milk teeth until they're ready to come through between the age of 6 and 12, and they push out those primary teeth. Well, why do we have this happening? Well, we live very different life now than our ancestors lived. Probably our ancestors had already ruined or used their primary teeth by the time they got to age where the permanent teeth emerged. So they were simply another set that we could deal with. However, these days we have much better healthcare. We take better care of our teeth. So we don't really have to replace them. It's just something that happens. Rarely do we have very damaged teeth by the time the secondary teeth come, the permanent teeth. But that's evolution. That's what we're dealing with. So in terms of studying the teeth, how I approach it is make your life simple when you can. You know I'm all about keeping things simple. I would recommend that you only learn 1/4 of the mouth. Because if you divide mouth in half, both horizontally and vertically, you'll notice that the order of the teeth is repeated in all of these sections. So if you learn only 1/4, you can just use what you've learned and mirror image it in any way that you have to learn other teeth. So let's go through them, and we're going to start here from the most anterior teeth, and then we're going to move towards lateral and posterior side. So we're starting at the front, and you can look at any of these quadrants that we see here. I'm just going to pick one that's easily visible for me because I actually have other screens on top of the slides as well since I'm doing the teaching. So we're going to start with incisors. So incisors are these two, more of a flat teeth, if you wish. They're very wide and very flat at the front. Some have described them to me like rabbit teeth, if you wish. Really, the good way to learn different kinds of teeth is by thinking of their function. These teeth would be ideal, for example, for biting apple, for example. So really getting into something, they're not really tearing kind of teeth, they're simply biting a bite out of something. So that's our incisors, 2 of them in each quadrant. Then the next teeth, 1 of them in each quadrant, is going to be our canines. Canines are sharp from their edge, especially on the upper jaw. And I think what we see in the media and with the celebrities is a little bit misleading. Really, canines that you see on most celebrities have been a little bit of sanded down. They should be quite sharp. If you wish, a little bit more like our vampires, what teeth they have. So they're sharp, they're ideal for tearing things like tearing meat. So that's probably telling us evolutionary that we have been also eating meat when our teeth have evolved for that. Then the 2 after that are going to be our premolars. Their function is something in between what we've seen before and molars. So they're kind of in between kind of a thing. And then we have our molars. And molars, if you look at their structure, their shape, that tells us about their function. They're especially well adapted to grinding food. So let's think, for example, taking a nut that you take to the mouth and you grind it down, or whatever you could end up grinding. So they're especially well adapted to that. So premolars are kind of semi-good for that. Not perfect, but better than the incisors and canines. So kind of an in-between type of teeth. So that's really what you need to know in terms of naming teeth in mouth. I would love to go in more detail, but in this review session, we're not able to do that. We're going to look at the tooth structure just briefly. And how we're going to review the tooth structure is by looking that there's two parts. There's the crown, which is the part that's above the gum. And then the part that's below the gum and in the jawbone is going to be our root. So let's start with the crown. And the outermost layer of the crown is our enamel. That's the probably the hardest substance in the human body. And we really don't want to damage that. We don't want to lose it because it's the layer that protects the rest of your teeth. So that's why all these toothpastes and other stuff highlight that, oh, this is good for your enamel. It fixes it or prevents the damage to it, stops damage to it. Underneath this layer, and notice this layer covers all of the teeth that's exposed to the outside, even slightly underneath the gum line, so that really other parts of the teeth should not be exposed if you have not damaged your enamel. The second layer underneath that is your dentin. Dentin is really kind of the bulk of the teeth. It's not as hard as the enamel, so you want to protect the outermost layer so you don't end up to this dentin layer because that can get damaged much more easily. And inside that we get our central cavity. That's where your blood vessels, your nerves, and your connective tissue is located. So if you have damage to your teeth and it ends up spreading all this way to the central cavity, this is when we're getting the really, really bad pain because the nerves here are exposed. This is also if you need to do the root canal, where we try to get the needle to, and then we actually kill or desensitize these nerves here. That's when you take that long needle during the root canal. And I've got some good stories. I'll try to have those included in the lecture videos of students who, when I was teaching, who ended up, for example, snapping the needle that's being used for a root canal inside the patient's tooth. There's really no other way of getting that snapped part than breaking the entire tooth. So that's kind of a little bit of a decision. Do we just leave it there or do we remove it? But that's a story for another time. In terms of the root of the tooth, we've got two layers there that we need to know. We've got a cementum, thin layer right against the dentin, and that really does what the name suggests. It kind of cements the structure where it should be. And then we've got our periodontal ligament, which is an anchoring kind of a ligament, holds the tooth in the socket within the jaw. So those are the parts of our tooth that we need to know. I hope that that is not too simple and it's not too complex. We're keeping it to the level. I'm trying to navigate between students who are going to this field and students who are not, so we have some balance in terms of how much detail we put in. Let's have a look what else we should know. Then after that, we end up with pharynx. So you remember behind the mouth, we had our oropharynx. And after that, we would have our laryngopharynx. There is, of course, the nasopharynx, but that plays no role in the digestive system. That's for the air only. So the oropharynx and laryngopharynx are shared both by the digestive system and respiratory system. So for both for food and air. What else should we talk about? We should talk about the oesophagus. And you'll notice that I always have trouble saying it like you Americans do, because in British English you would put an O in front here. So depending a little bit where you've studied, how you end up spelling those. But I try to do as much American as I can, but forgive me for sometimes forgetting that. So oesophagus is an interesting structure. because it is nothing else but a muscular tube as part of the digestive system. It does no absorption whatsoever. It does no digestion whatsoever. It simply transports food from your mouth to your stomach. And that's all it does. It's about 25 centimeters long. And really, like I said, its only function is the transport. What is interesting, though, is since it is a muscular tube, it moves the food doing that peristalsis, that movement that we saw earlier on. I don't know, I might have to go quite far back. This movement here, the wave-like movement of food along the digestive tract. So your esophagus moves material using this peristalsis because it is a muscle tube. And what's interesting that even if you were standing on your head, you're still able to transport food or water from your mouth to your stomach, thanks to the peristalsis of the esophagus. And you can try that if you're bored, or if you have a friend who you want to recruit to help your anatomy and physiology studies. It is true, esophagus allows us to transport this food. It's not the gravity that transports the food from the mouth to the stomach. It's actually this wave-like movement, this peristalsis of the esophagus. That's why you can swallow food also in space, liquids. They travel where they're supposed to go. Esophagus can break through the diaphragm. There's only few structures that go through the diaphragm. You remember that kind of thin, broad muscle layer that separated your thoracic and abdominal cavity. Esophagus punctures that, also your aorta, your ascending aorta, your inferior vena cava punctures that, but not a lot of other structures. So esophagus only has very few openings there, or very few holes, or I mean, diaphragm has very few openings there, and esophagus was one of those. What I want to show here, and we've already discussed about the bolus, what I want to show here is a microscopic image of esophagus. What's interesting, what we notice here, that it is actually shrinked or closed at all other times when we actually follow food. That's the only time that it expands wider. And that's because we want to leave as much space in this area for the trachea to be open, so to secure the airway as possible. Other thing that we can notice here on this stained microscopy image or sample of the esophagus, we can notice those 4 layers that we had. We can see our mucosal layer, kind of this more purple. Then we can see the submucosa, this bluish layer. After that, 2 muscle layers that helped us to do this peristalsis. And then our adventitia at the very far end. So we're really seeing all these that we saw before when we were studying, and we keep seeing them at different parts of the digestive tract. Notice though that with the esophagus, this mucosa doesn't allow any materials to pass, to be absorbed, because esophagus didn't do any absorption. It simply transported the food, but still we see this layer. I think that that's everything that I need to say about esophagus. The next is kind of physiology and ties everything together. Concept of deglutition. What is deglutition? Deglutition is a fancy word for swallow. And what happens when you swallow? This is probably quite obvious for most of us. Our tongue is used to push the food a little bit to the back of the throat. Your soft palate and uvula rise up, sealing off the nasal cavity. We also get our epiglottis moving up, so closing off the respiratory tract, closing off the trachea. Your throat kind of jumps up a little bit to catch that bolus. I should have in our lecture material a nice video of that I took during my time in the medical imaging of how the throat actually jumps to catch the food, the bolus in this case. And then it continues down the esophagus as peristalsis. I can't see the last picture because I've got another screen on top of it. But the question that we can ask about deglutition is that, is it voluntary or is it involuntary? And that's a good question. Well, actually, the first phase of swallowing is voluntary. You can choose to swallow or you can choose not to swallow. However, once you have started the process of swallowing, you cannot stop it. So the later part of that is involuntary. So it's kind of mixture of two. That would make a fun exam question. So that's all that I can think of that we would need to know about the esophagus and swallowing. And that takes us to talk about stomach. And stomach is this kind of J-shaped pouch, about 25 to 30 centimeters in size. Notice that the size of the stomach changes a lot. We often see it drawn in diagrams as something like this, but often your stomach is not this full. It's actually much more shrunken, much smaller. Located below the diaphragm on your upper left part of the abdominal cavity. I think that that's a good start for what we need to know about stomach. Important thing to notice at this point is that now we had food. When we mixed the food with saliva and mechanically broke it down, it turned into bolus. And now once the bolus reaches stomach and gets mixed with the stomach acids, it stops being a bolus and becomes chyme. Chyme is this liquidy stuff. It's much more liquid than anything that we've seen so far that the food could be. So if you've ever thrown up, that's chyme kind of structure or material that comes up. So that gives you an idea of what it is like. Stomach can change much in size if you look, from being very, very small to very large. But most of the time, it's not this extended. Inside the stomach, as we will see later in one of the pictures, there's these little folds called rugae. They are what allows the stomach to be stretched. So when stomach is empty, you find these little bumps, rugae in there. And I would love to have face-to-face classes with you because I normally bring at least a fetal pig or a rat, and we dissect that and take their stomach and cut it open. Sometimes that can be a bit of a stinky surprise, but once you open that, you actually see those rugae there. Let's have a look at different parts of the stomach. So stomach was just a temporary storage tank. There's very little absorption that happens there. That's mainly a storage site, so no absorption really there. It's too acidic for most of the digestive processes to actually take place. It's just a holding tank, if you wish. So let's have a look what parts of the stomach we have. And remember, this is a diagram of a stomach that's totally full. We rarely see this as it is, but you'll see some pictures on the full lecture material that I've taken of real patients. So here's our esophagus coming down. And the part where esophagus joins stomach, we have as part here, and we call that part where this junction of esophagus and stomach takes place, or the first part of the stomach that's right after the junction, we call it cardia. So cardia is where this junction between stomach and our esophagus is present. I'm just looking what my cat is doing, if you're wondering why I'm staring there. The next part is fundus. You'll notice that fundus is higher up than where your esophagus joins the stomach. So this is important structure to know. This is where gases, air usually ends up building up. So again, when you're taking medical images, you might end up seeing air bubbles. The air bubbles are going to be at the fundus of the stomach. And the majority of the stomach is the body of the stomach. And right at the distal end of the stomach, we have an area called pylorus. This can be divided into pyloric antrum, more proximally and more distally as pyloric canal. But as long as you know that it's pylorus, I'll be happy with that. You don't need to know in more detail. And after that, we get to the small intestines. And we'll talk about small intestines a little later. But what we need to know is that we've also got a couple of sphincters that control these openings from the esophagus to stomach and from the stomach to small intestines are not open at all times. Well, first of all, we've got lower esophageal sphincter. So where the esophagus ends and stomach connects, there's a sphincter. The function of the sphincter is to prevent stomach acids from climbing up to your esophagus. What happens if stomach acids climb up there? You get something that we describe as heartburn. So that's the stomach acid irritating the esophagus. Try not to rip my notes. Irritating the esophagus. So it's not really nothing to do with the heartburn, but that's what's happening there. And if this happens, this can be due to many things: excessive food or drink, obesity, pregnancy, running. There's many reasons. But if this becomes a prolonged thing that you often have heartburn, you really should get it addressed. Because if you constantly keep getting stomach acids to your esophagus, you can end up with ulcers in there. And these ulcers, not only they're not pleasant, but if these ulcers keep getting irritated, they end up getting a higher risk of cancerous growth starting to take place here. So that's kind of the announcement that I want to make about it. Don't let yourself suffer from heartburn. Get it addressed. The interesting thing is that this is not really an anatomical sphincter. If we cut into the human body, we likely don't end up seeing an actual sphincter there. It's more of a physiological sphincter than an anatomical structure. Well, at the other end, where our pylorus joins with the small intestines, we have pyloric sphincter. And this is actually an actual anatomical structure that we can find that separates the stomach from the small intestines and controls when do we let the stomach contents continue to the small intestines. Anyone who has had the runs knows that that's when there's no control. That's just everything gets dumped into there. So we want to control that we store food there until small intestine is ready to deal with that food. So stomach holds it there. But sometimes this sphincter can be broken. What else should we talk about? We should talk a little bit about the hernias. But before we do, I want to show two more structures about the stomach. This more medial border, you see like a curve here. This dotted line is just referring to this. I just couldn't fit it in there. This is known as our lesser curvature. And on the more lateral side, the bigger one, we call that greater curvature. So those are terms that we can also use when we describe locations within the stomach. So terminology that we might want to know. So lesser curvature and greater curvature. I said that let's talk a little bit about hernias. So hernia can take place in many different parts. In this context, we're looking at the stomach. So here we see our diaphragm, this muscular sheath, separating the abdominal cavity from the thoracic cavity. And there were only very few holes that went through our diaphragm. And one of those was the esophagus. Here, stomach is actually squeezing up through this hole to a space where it shouldn't be. It shouldn't be in the thoracic cavity. So that's just something to know. That could be a problem. There's many different kinds of hernias on this area as well. It can be an issue. It can also be totally asymptomatic. But I just wanted to mention it, just so we know one clinical condition. Let's look at the stomach a little bit more on this diagram. What we see here are these three layers. We had seen the circular layer, which was the innermost, and longitudinal muscle layer, which was the outermost. But in stomach, we've also got this oblique layer, so the third muscle layer. And that was because stomach needed to be able to expand and contract, mix the food, break down the food. We also see on this diagram the rugae, these little folds inside the stomach. So that's something to know about as well. And these folds were the ones that allowed, again, the stomach to shrink and expand. When you don't have food for a long time in the stomach will actually physically shrink. If you don't eat other than just tiny meals, your stomach will slowly get physically smaller. Then you can't go and eat a crazy big meal all of a sudden because your stomach can only stretch so much. Well, if you constantly eat big meals, your stomach will keep growing bigger and bigger, expanding bigger. So this is really where it becomes an issue of lifestyle choices. And there's all sorts of things, like we could talk about stomach when you put a gastric band and all those things. But that's for the full lecture on the lecture videos. So let's have a look at the secretions of the stomach quickly. We won't spend too much time on this. We have... When you look at them, I'm going to have to move some of the screens so I see the same stuff that as you see. When you look at the stomach and the lining of it, stomach is lined by simple columnar epithelium. So these column-shaped or pillar-shaped cells of the epithelial cells. Within those, we've got structures known as gastric pits. These are these depressions between these cells. And at the very bottom of a gastric pit, we've got the gastric glands that produce the gastric juice. And gastric juice is the secretion of the stomach. You remember this part did not do much of absorption. It's very acidic. And because of the acidity, most enzymes, most digestive processes don't really work that well. There's not that much happening of absorption-wise within the stomach. How much gastric juices do we secrete in a day? It's actually a huge amount. It's 3 liters a day. So it would be maybe 1.5 of these. So that's a lot that we secrete in a day. So we're going to have a look at these stomach secretions. It looks like a huge list, and you might be like, oh my God, that's a pain. But remember that in the exam, you can use your notes, you can refer to those. And really, once we go through these, it starts making sense because these are all connected in one way or another. And we're going to start with the pepsinogen. Pepsinogen is secreted from the chief cells. It's inactive. But once we get hydrochloric acid, so stomach acid, which is secreted from parietal cells, that activates pepsinogen, which then becomes pepsin. So pepsin is the active form of pepsinogen, and it gets activated within the stomach when we mix it with the pepsin again, we mix it with stomach acids. Now that it's activated, it starts breaking proteins. So proteins are broken down, digested within the stomach. Most other stuff isn't digested in the stomach, but proteins are the one thing that actually get broken into smaller within the stomach. Thanks to our active form, pepsin, which is made of pepsinogen when the stomach acids are present. What else should we know? Well, we should definitely know our gastric lipase. Lipase tells us that it breaks fats, lipids. That's again secreted from the chief cells. But gastric lipase doesn't work very well within the stomach. It's because this pH, the acidity within the stomach, makes the gastric lipase very weak. It doesn't work well. It's only once these materials leave the stomach that the pH neutralizes. It's no longer as acidic in the small intestines when the gastric lipase can start working really well in breaking the fats. So the proteins were the one exception that got digested in the stomach. Most others digested only after the stomach in the small intestines. What else? Stomach secretes a lot of mucus that really protects the stomach wall lining. That's secreted from the goblet cells and mucus glands. Really, there's a lot going on. It's very acidic, so this mucus protects that we don't end up digesting the wall itself. And of course, we also secrete something known as intrinsic factor from the stomach. And this is what plays important role in vitamin B12 absorption and is secreted from the parietal cells. So that's a really quick review of the stomach secretions. Here's a clinical relevance that I want to mention. You can have ulcers, stomach ulcers. So there's sores in the walls of the stomach. These can cause all sorts of risks and concerns for us. If there's the damage to the wall, we can of course get inflammation and bleeding. This is a great way for bacteria to spread. Certain drugs such as aspirin can cause gastric ulcers. So we want to follow the amount of dosages that we say, how much aspirin you should take, so you don't end up with gastric ulcers, stomach ulcers. Well, I'm not going to go into detail about the regulation of the gastric secretions. That's mainly for the full lecture video. I just want to show you that the first phase is simply a reflex when you see, taste, smell, or think about food. Then later stages take place more further down the line, later on in terms of the time. There's stimulation and there's inhibition depending on the amount of pH that we have there, and also depending on how much we're stretching the wall of the stomach. I will check my emails, okay, after the class. So how much we're stretching the stomach, how much we're stretching the small intestines, large intestines. So that's really all that we need to know about that topic. The next thing that we need to ask is why stomach is not that good in this absorption. I've mentioned that multiple times that no, we don't absorb things well from the stomach. Why doesn't it do it? Well, it's because it's such an acidic environment. There's no villi. We had the rugae, but they're much bigger than villi, and there's this mucus layer. So it's really hard for the stuff to actually get across this membrane. So most of this takes place actually within the small intestines. I'm trying to think, is there anything else that we should mention about this? I can't think of. And to wrap up this part, and the last part is really, really quick, I just want to show you that we've moved now with the terminology. Food was what we ate. Once we mixed it with saliva and physically broke it down, we started talking about bolus. Once it enters the stomach where it gets mixed with stomach acids, it becomes chyme, and then it becomes feces at the large intestines where we reabsorb most of the fluids, most of the liquids from this really liquidy stuff. And we're only left with the stuff that's really undigestible. So we're going to have a look of the small intestines. This is going to be much quicker. So small intestines start at the last part of the stomach, which was pylorus, and the first part of the small intestines is ileum. So it starts at the pyloric sphincter, and the first part was duodenum, and the last part of the small intestines is ileum. And ileum then turns into large intestines, and the first part of the large intestines was cecum. So we have an ileocecal sphincter there. We'll have a look at that later. But it is the small intestines that are the major site of absorption and also digestion. So what are the parts of the small intestines? We've got three parts: duodenum, jejunum, and ileum. And you're going to be like, oh my God, how do I learn those? How do I remember those? You remember I'm all about silly mnemonics. I remember the order of duodenum, jejunum, and ileum by thinking of this guy whose name is DJ Ill. He plays really sick music. So, DJ Ill, duodenum is the first part of the small intestines, right after the stomach. About 25 centimeters long, just curves around the pancreas. You'll see pancreas here, and we'll mention that quickly. So that's the duodenum, the first part right after stomach from here. And you'll notice there's openings we'll talk about then later. After that, we've got our jejunum, And then we've got our ileum. Where does jejunum end and ileum start? No one knows. There's no clear definition. But we know that the middle part of the small intestines is jejunum and the last part is ileum. So let's look at the small intestines in terms of their wall. There's a lot of folds, there's a lot of villi, and there's a lot of microvilli. All this allows us to have a lot of surface area for absorption. Actually, if you spread out the surface of the small intestines, it's about the size of a tennis court. So a lot of surface area. So it's ideal for stuff to cross into the body. And some of the secretions from the small intestines, we've got mucus from the goblet cells, peptidase that broke down the peptides into amino acids, sucrase, maltase, lactase, we've heard about those before. They break disaccharides into simple monosaccharides. And lipase, we already talked about that broke the fats into fatty acids and glycerol. So then we get to the last part that we need to know, large intestines. Large intestines form kind of this frame around the abdominal cavity. So it's on the lateral aspects and on the superior aspects of the abdomen. And the first part of the large intestines is actually cecum. So this is where the end of the small intestines, which was ileum, and the start of the large intestines, which was cecum, come, and the junction between those two is ileocecal sphincter. We have also from the cecum an appendix hanging out. And we talked about appendix already earlier on. That really serves no purpose these days. It's simply an evolutionary remain from when we ate more of plant-based food. Bacteria lived there, but that can get inflamed. You can have your appendix removed as well. This is an appendix during a surgery. We're removing it. And you'll notice that we're able to do surgery these days that leaves very little scars externally. So appendix surgery is not that unusual. We have high success rate on that. After that, we get to the colon, which is the most prominent part of the large intestines. Colon can be divided into 4 parts. We have the ascending colon that climbs up, the transverse colon that crosses across at the superior aspect, descending colon going downwards, and then sigmoid colon, which you'll remember because it makes a little bit like a shape of an S. And at the here's a picture of the colon. You'll notice it's much larger. We can see the appendix here. This would be cecum. This would be the ileocecal opening. A little bit of fat pads there connected by our peritoneum. You'll notice there as well, mesentery. And at the very end, we've got our rectum that stores until we're ready to get rid of the feces. So the main function of the large intestines is not absorption. The main function of the large intestines is the reuptake of water and also electrolytes. So this is where the chyme gets dried up and becomes now feces. You'll end up finding a lot of flora, so bacteria growing on the walls of the large intestines. That's mostly beneficial flora. A lot of fermentation, vitamin synthesis takes place there as well. And the amount of flora on the walls of the large intestines means that it's all covered with some sort of bacteria or other forms of life. So pathogens from outside cannot really establish themselves there. There's simply no room. This is why if you do a lot of colon cleansing and things like that, you wash away the beneficial flora, you are actually more susceptible for getting issues with pathogens entering. And the very last part that we're going to cover on this review is going to be super quick. I'm just quickly going to refer to these accessory organs that we've seen before. We've already talked about the salivary glands, so we've only got 3 left. We're going to talk about liver, gallbladder, and pancreas. So let's go through them quickly. Liver is really a large gland that we end up finding in the body. It has 4 primary lobes and different structures that then divides those. Those are going to be addressed in the full lecture video. But what I want to highlight, the liver has a lot of functions. Liver, I started listing them one day and I got to 100 and I gave up. But really, the big function of the liver is to produce bile. And don't get confused. A lot of the time, people who have done a lot of A&P, even nurses, get confused. They think that bile would be produced by gallbladder. That's not the case. It's the liver that produces the bile. And then the bile travels to gallbladder to get stored and concentrated. Here we can see the falciform ligament, round ligament separating the lobes of the liver. Remember, these are all averages. We actually have a little bit of variation. Sometimes we find livers with more lobes, with less lobes. In terms of the bile, bile was produced in the liver and then it travels down the common hepatic ducts. About halfway through down, where the cystic duct carries the bile to the gallbladder. Bile is stored there. During its storage, it also gets concentrated. And when you eat fatty food, your gallbladder releases the bile back down the cystic duct and then the bile duct to the small intestines, to the duodenum. And this is why we broke down the fats in the small intestines. If you have your gallbladder taken away, you really shouldn't be eating fatty foods because it's going to give you the runs. You don't really end up breaking the fat. So really, gallbladder, the main function, the storage and concentration of the bile. And I'm just trying to get an axis there. That's the main function of the gallbladder that I want you to know. Let me change this to here. And sometimes you can have something known as gallstones produced. That's when there's cholesterol that escapes the bile and ends up forming crystals inside the gallbladder. So these can become an issue and we might need to deal with it. There's many different ways, such as ultrasound, sound, so even surgery at the worst place, or certain drugs can help. We don't want to have gallstones. And the last one that we need to talk really quickly is our pancreas. You remember pancreas was this kind of this might be a bad description, but a carrot-shaped structure. It has a wider part and more narrow part. We also remember that pancreas was both endocrine and exocrine. So when we talked about endocrine glands, when we talked about hormones, that was all to do with the sugar regulation through the insulin and glucagon. But in this context, our main interest will be on the exocrine function of the pancreas. So pancreas produces pancreatic juice. And this pancreatic juice is important for us because it is the most complete digestive juice produced in the body. So that's really what does the main job of breaking, digesting materials. And that actually gets released to the small intestines, to the duodenum, through the same hole where we release the bile from, through pancreatic ducts. And here we see the opening, that little dot there. And that really is the main function of the pancreatic juice is to neutralize the secretions that come from the stomach that were very acidic, and that allows the absorption to start, and that's why most of the absorption happened in the small intestines. And that really concludes our review. I know that this took way longer than I had intended, so that's all we have to know about the digestive system. You'll notice the emphasis is in the digestive tract, and we'll talk a little bit about we talk a little bit about the accessory glands, but that's not the main emphasis on our on our review. And I'm just going to jump back before we finish to the slides that we had for this review session. Remember that you're going to have full lecture videos that cover everything, but we've covered pretty much the main points on this review session. And just jumping back there to our review PowerPoint, we got to the fact that we reviewed the key points of the digestive system. Like I said, if you need additional assignments, if you can't use Connect anymore, I'm happy to provide those. But do contact me so I know which chapters, with knowledge which chapters you need those for. But all the Connect work is now open if you want to make advanced work on that. Like I said, if you're missing any assignments, any exams, any practicals, just let me know. I'll be happy to make sure that you have a chance to complete those. The finals project, we'll talk about that later on. One question that came up during this morning session was that is there going to be a separate extra credit activity? I would prefer that you can gain your extra credit as part of the finals project. So there's a maximum amount of points that you can earn in finals, but then you can earn extra points beyond that maximum amount if you do even more work on that. So that's the finals project. But even if still you're not where you need to be, then I'm happy to come up with an extra credit activity. But I try not to have too much activity for you, and I try not to have too much for me to grade. And I think that pretty well covers everything that I wanted to mention. Just to jump back, because we noticed a couple of things as we started this session. So like we discussed, there was an issue with the practical tree. So practical tree, I'm going to have to do something about it, and I'll let you know whether I'm able to rebuild it. And if I'm not able to rebuild it, then we're probably just going to have every student getting full marks for that automatically. I'll have to think about it overnight, what I'm going to do, but I'm going to be sending an announcement about that to the entire class. But make sure you get the exam tree done if you haven't done it. I'm saying it for the recording. I don't help you. This is the last week to finish that. And make sure you check your exam 2 and practical 2 grades. They should be available. If you haven't got the practical 2 grade available, it's available by the end of today. Make sure that you review those. Also, if there's any issues with those, if I've written somewhere that there is, thank you, have a good night. If I've written there that, hey, you've got full marks and then you didn't get those, let me know. I make mistakes as well. And the critical thinking, I'll get that back to you soon. Make sure to do both labs for this week. And that's all we have for tonight. I hope you have a good night. Thank you for joining me. And I will post an announcement with the answer, what we're going to do with the practical tree. Have a good night. Thank you for joining me. I'll make sure that you get extra credit for having stuck around this long. I know that it was a long night. Good night.
Good night. Bye.
Bye. And for the recording, I just say that I think that we covered everything. If there is anything that you have questions, you can always message me on Canvas. I recommend the Canvas messages rather than the email. The email gets really crowded really often. So that's all for this review session. I was hoping to keep it nice and short, but it actually did take a little bit longer than I had intended. Pretty much the same as the morning session. So that's the end of the week 13 recording, and I hope you have a good week. Let me know if you need any help or if there's anything I can help you.